Okay, good morning everybody. Are we ready? So let's get started. And today we still going to talk about lipoproteins, and we've already finished the chylomicron VLDL metabolism, right? And also LDL. And we know that the chylomicron and VLDL are primarily triglyceride-rich lipoproteins, right? And the primary role of these two lipoproteins is to transport triglyceride either from diet or from the liver and to the adipose tissue, of course, during the fat condition, right? And for VLDL, which is synthesized in the liver, can also transport fatty acids, which initially from adipose tissue during the fasting to which tissue, you guys remember? Skeletal muscle, right, during the fasting, and for utilized as energy of fatty acids. And HDL and LDL primarily are the cholesterol-rich, particularly cholesterol ester-rich lipoproteins. And HDL usually we think is good cholesterol, right? And LDL is a bad cholesterol. And the reason is the LDL basically transported cholesterol or deposit cholesterol into the peripheral tissues, and HDL basically is transport cholesterol from peripheral tissue to liver and dispose it, right? So these two different type of lipoproteins regulate the balance or homeostasis of cholesterol. It's very important. So now we're going to talk about HDL. So what are the core lipids in HDL? Yeah, cholesterol esters, and some of also lipids are the triglyceride, right? And apoproteins on the surface of HDL. And ApoA1, again, is unique to HDL, right? Means that ApoA1 is only present on the surface of HDL. And HDL also has the ApoA2 apoproteins. And whereas HDL synthesize liver and intestine, right? So liver and intestine are the primary sites for the synthesis of HDL. And what are the functions of HDL? We already talked about it, right? So there is very important role, so-called reverse transport of cholesterol, mean usually transport cholesterol from peripheral tissue if we have a lot of cholesterol that we don't need that much, then the excessive cholesterol need to be disposed. So HDL plays this role to transport excessive cholesterol from peripheral tissue to the liver, and in the liver, cholesterol being synthesized as bile acid and excreted. So this is the primary role of a HDL, and HDL also plays another role, is that the transfer ApoC and ApoE to the other lipoproteins, for example, chylomicron and VLDL. Remember the chylomicron and VLDL, when initially synthesized in the small intestine or liver, is so-called nascent, right? The chylomicron means that these two chylomicron either do not have an ApoC or E, or not enough ApoC or E, so they receive the additional ApoC and ApoE from HDL, right? So this is another function of HDL. And reverse cholesterol transport. Means that transport cholesterol from peripheral tissue back to liver. And this reverse transport process actually is accomplished by so-called HDL cycle. So HDL cycles involve the two different types of HDL, alpha HDL and pre-beta HDL. So alpha HDL and pre-beta HDL, they have different composition of lipids, right, as well as apoproteins. So it means that these two different types of lipoprotein can be converted, each other and change the composition of lipids to complete the transport of cholesterol. So let's look at the more, we can watch the video later. So let's look at the more specifically details about the HDL cycles, right? So alpha HDL basically is to give the cholesterol ester to the liver, right? So alpha HDL contains lots of cholesterol esters, right? So it's the final version of HDL. So alpha HDL get enough cholesterol, form the cholesterol esters, and then give this cholesterol ester to the liver. So that means the last, the alpha HDL is picked up by the liver, right? And pre-beta HDL receives free cholesterol from peripheral tissue and forms alpha HDL, right? So let's look at the composition of these two different types of HDL. So composition of pre-beta HDL. So pre-beta HDL basically is as a no or little lipid core, right? So it's they basically has just the surface phospholipids and ApoA1. Do not have enough or a lot of cholesterol in the core, right? So the poor lipids, the pre-beta HDL. And alpha HDL, in addition to the surface phospholipids and ApoA1, it also has ApoA2. And the most important thing is the alpha HDL has a lipid core, right? This lipid core contains a lot of cholesterol esters. And it also has ApoC2 and ApoE, right? And the other thing, before we talk about more detail about the cycles and important enzymes that catalyze the whole process of reverse transport, the enzyme and proteins you guys need to remember, right? The first one is called ABCA1, which is the ATP binding cassette transporter A1, right? So ABCA1 is the transporter. And the function of ABCA1 is to transport free cholesterol from peripheral cells to the beta pre-HDL, right? So ABCA1 basically is located on the surface of the peripheral cells, right, to help load free cholesterol from peripheral cells onto the pre-beta HDL, right? And a second protein or receptor is called SRB1. SRB1 scavenger receptor B1, right? It's located on the liver cells. And this receptor is similar to LDL receptor, right? The function to pick up the LDL. And LCAT, the lecithin cholesterol and isotransferase, And this enzyme present in the plasma, in the circulation, right? And the other protein or transport protein enzymes is CEPT, is cholesterol transfer protein, right? And this CEPT proteins also localized in the plasma. So two enzymes or proteins that are present in the plasma, right? And ACAT is called acyl-CoA cholesterol acyltransferase, and this enzyme is also located in the cells, right? So I want you guys to try to distinguish these two different enzymes, LCAT and ACAT. So LCAT and ACAT, they have a same enzymatic function, right? They have a same function that is catalyze the formation of cholesterol ester from free cholesterol, right? In other words, these two enzymes have the same function. Their function is to catalyze the esterification of free cholesterol to cholesterol esters, right? But they have a different location. LCAT is in the plasma circulation, and ACAT is inside of cells, right? They have same function but different locations. They catalyze the same reaction, that is esterification of free cholesterol to cholesterol esters. Okay, so what happens to alpha HDL when it becomes beta HDL, right? So alpha HDL we know have a lot of lipids, right? Core lipids, cholesterol esters, and beta, pre-beta HDL basically is at the poor lipids, right? Almost no or little lipids in the core. So the transition from alpha HDL to beta HDL means that you have a lot of lipids that are being transferred out of alpha HDL, right? And we also mentioned the alpha HDL basically is picked up by the liver. So the liver picks the lipids, particularly cholesterol esters, inside of the alpha HDL and makes alpha HDL become lipid poor pre-beta HDL, right? So the things happening to the this transition is that first of all the reduction of HDL core volume means the loss of the cholesterol esters, right? Basically picked up by the liver. And the second thing is the dissociation of ApoA1 with alpha HDL. So it means that ApoA1 is also lost, right, from the alpha HDL. And internalization of cholesterol from alpha HDL basically by the liver, right? Liver picks up the cholesterol esters in the alpha HDL. And the hydrolyzed triglyceride in the alpha HDL by hepatic lipase, right? So alpha HDL also contains the triglyceride. Right? Also, more lipid are the cholesterol esters, but still also has some small amount of triglyceride. So both type of lipids eventually being picked up by the liver. Okay, now what happened to the pre-HDL when it's become or converted back to alpha HDL? So alpha HDL is more abundant in the circulation, right? They get lots of lipids from the peripheral tissue, lots of cholesterol from the peripheral tissue, so eventually picked up by the liver, right? And pre-beta HDL which is synthesized initially in the liver and be transported to the peripheral tissue. So it's very concentrated in the extra fluid of the tissues, right? So it means that the pre-beta HDL is more concentrated in the tissue, in the peripheral tissue. So, so pre-beta HDL is enriched in the extracellular fluid. Yeah, we all talk about this already, right? And accepts or receives the free cholesterol and phospholipids from peripheral tissue, peripheral cells, right? And this process, ABCA1, we just mentioned a couple of slides earlier, the ABCA1 is the transporter, right? The free cholesterol transporter. So it's very important, the ABCA1 on the surface of peripheral cells that transport free cholesterol from the cells and load it to the pre-beta HDL, right? So pre-beta HDL basically receive enough free cholesterol of phospholipids from the peripheral cells, tissues, and then forming the so-called discoidal HDL and re-entering the plasma, right? So, so in the plasma, there are also several reactions and happening, we're going to talk about later, that to eventually to make pre-beta HDL converted to alpha HDL, which is rich in the cholesterol esters and triglycerides, right? So now what happens to alpha HDL within the plasma or in the circulation? And we mentioned this enzyme, right, LCAT. This is enzyme that catalyzes acidification of free cholesterol, which initially picked up from the peripheral cells, right? And to form the cholesterol esters, right, in the circulation. And once it's formed the cholesterol esters, that cholesterol ester being internalized into the core of the alpha HDL. Free cholesterol usually present on the surface, right? The surface, the monolayer of the lipoproteins. So once it's acidified to cholesterol esters, then enter the core, lipid core of the alpha HDL. And ApoA1, which is unique to alpha, to HDL, and its function is to activate LCAT, right? And there are also things happening in the circulation, and it's called exchange of a cholesterol ester, the triglyceride, between HDL and the other lipoproteins like LDL, IDL, and VLDL. Right? So by this enzyme of transfer proteins called CETP, so CETP is the transfer protein, transfer lipids, right, for the exchange between HDL and the other lipoproteins. And this is the receptors on the liver, liver cells, and can mediate the pickup of the rest of the cholesterol esters, right? And inside of the liver cells, once the HDL, alpha HDL be picked up, the cholesterol inside of HDL being used for bile acid or steroid hormone synthesis, right? Okay, so too much kind of description text, right? So now I am going to show the diagram to summarize what I just described, the reverse cholesterol transport or HDL cycle, right? See how HDL transport cholesterol from peripheral tissue to liver for the disposal. So again, the pre-beta HDL is the lipid poor lipoproteins, right? And initially synthesized from the liver and can be transported to the peripheral cells, right? So this left side just represents the liver, right? And right side, the peripheral tissue cells. And this is the membrane, the cell membrane of a peripheral cells, right? And in the between, this space represents the plasma, right? So all this reaction actually occurs in the plasma. Okay, so pre-beta HDL does not have much lipids, initially synthesized in the liver and transported to the peripheral tissue, right? And then picks up the free cholesterol, FC, and phospholipids, PL, right? And then uploaded to the pre-beta HDL become a little bit lipid-rich, the HDL called discoidal HDL, right? And again, the ABCA1 is the transporter of the free cholesterol as localized on the surface of a peripheral cells, right? It's very important, the key transporter that control the uptake of a free fatty acids by beta HDL, right? So if something happening that ABCA1 either not functioning well or deficient or mutation, then this pickup will not be happening, right? So then you have a lot of free cholesterol cannot be picked up by pre-beta HDL, so lots of free cholesterol being actually accumulate in the peripheral cells, right? So ABCA1 is important transporter proteins. They are actually of a case in the patient, ABCA1 deficient, so have some problem with the pickup of cholesterol or disposal of cholesterol. So a lot of cholesterol accumulate in the peripheral cells. If it's in the macrophages, the artery wall, they were causing atherosclerosis, right? So, and pre-beta HDL pick up free cholesterol and phospholipids and become disposal HDL. Then we entering to the plasma circulation, and in the circulation there are also several steps take place. First of all is the acidification of free cholesterol to cholesterol esters, catalyzed by this enzyme, remember LCAT, right, and present in the plasma. So once it's esterified, then cholesterol esters then enter this core of alpha HDL. So once they receive the lipids, then beta HDL become alpha HDL, right? So alpha HDL is the lipid-rich apoproteins. And the other thing happening is that exchange of lipids between alpha HDL and VLDL, LDL, and LDL. So basically, the 30% of cholesterol esters being given to the HDL, VLDL, LDL from alpha HDL. And in the meantime, the triglyceride being transferred from VLDL, LDL, LDL to alpha HDL, right? So eventually, alpha and HDL become still have a cholesterol ester rich, but it also have more triglyceride, right? If compared to the the initial beta HDL or HDL discoidal HDL, right? So finally, the alpha HDL, which contains a lot of cholesterol esters and some triglyceride, and picked up by the liver and mediated by these receptors, right? And cholesterol esters being utilized for the synthesis of bile acid, which is excreted, or can also be utilized for synthesis of steroid hormones in the liver, right? And once the cholesterol esters, the triglyceride pick up by the liver, and alpha HDL then become lipid poor pre-beta HDL again, right? And this will again go to the peripheral tissue and pick up cholesterol, and again, go the cycles, right? Any questions about this HDL cycles or the transporter, reversal cholesterol transporter, right? Okay, so let's watch some video. I think it's interesting and important, maybe help you to better understand the lipoprotein, metabolism. Okay. Okay, so this is the last slide for this unit. I just wanted to remind you guys what you guys need to know about the lipoprotein. First of all, the major components and function of 4 lipoproteins, chylomicron, VLDL, LDL, and HDL, right? Particularly the lipid content in the different chylomicrons and what the function. Some of the lipoprotein, for example, chylomicron and VLDL, you can think about in the fat condition versus fasting condition. Particularly for VLDL, actually can transport a triglyceride in both fat and fasting condition, right? But just the destination is different and the purpose is different because in the fat condition, right, the VLDL basically transport triglyceride to the adipose tissue for storage, and during the fasting, VLDL transport triglyceride to skeletal muscle, right, and for the energy. Skeletal muscle utilize fatty acids. And the second thing is the metabolism, right? So you probably wanted to enjoy, like your own the whole pathways, how, for example, chylomicron is metabolized. First synthesized in the small intestine as the nascent chylomicron, and receive the ApoC and E from HDL and become remnant, become mature chylomicron, right, and to the adipose tissue, and the enzyme important secreted from adipose tissue that can hydrolyze triglyceride in chylomicron, right, which is, remember, LPL, right? So and the pick up, adipose tissue to pick up fatty acids and reacidify back to the triglyceride and store in the adipose tissue, and the chylomicron remnant then eventually picked up by the liver, right? And this pickup requires which lipoprotein? You remember? The chylomicron remnant picked up by which apoprotein? Sorry, which apoprotein is required for the pickup of chylomicron remnant by liver? You watch the video and also we talk about it, right? ApoE, right? So it's important. And then VLDL, right? So you also try to start from liver, that VLDL synthesized the liver, and you also need to know what type of what sources of fatty acids that being synthesized to triglyceride packed into the VLDL, right? One source is of the diet, right, from the diet, and the other source is de novo synthesis, fatty acids initially from dietary carbohydrate, right? And the third source is of fatty acids in the triglyceride in VLDL is from the pickup from the circulation fatty acids, right? And this process actually is different in the physiological condition. Under the fasting, we have high levels of fatty acids, right, being released from adipose tissue. And also you need to note under the pathological condition, like a diabetic condition, right, and you also have high levels of fatty acids. So liver picks up and synthesizes triglyceride and packed into the VLDL. So this different sources of fatty acids you also need to know, right, in the physiological versus the pathological condition. And LDL, HDL, basically, try to understand the bad, good cholesterol. Right? The purpose of LDL is deposit cholesterol into the peripheral tissue or artery wall, and HDL is transport the cholesterol from from peripheral tissue back to the liver, right, to dispose the cholesterol. So this, just try to remember this whole metabolic pathways of lipoproteins, right? And the role of LDL receptors, which is very important, right, for pickup of LDL cholesterol, and present in which tissue is the most abundant, the most highly expressed LDL receptors. No one knows. I don't believe that. Liver, right? Liver is expressed a lot of LDL receptors. And LDL receptor number on the cell surface determine the cholesterol levels, LDL cholesterol levels in the circulation, right? Remember we talked about this, right? Because it controls the pickup, the LDL cholesterol into the cells. So the LDL receptor numbers is most important factor that regulates the circulating LDL cholesterol levels. And the factors that involve the cholesterol, the reverse cholesterol transport, the HDL cycles, ACAT, LCAT, CEPT, AB, CA1, SIB1, right? There's different proteins, where they're located, and what the function of these proteins, right? And one more thing, you need to distinguish the functions of LPL, hepatic lipase, and HSL. So what is the difference between LP and HSL? In terms of, first of all, the location, where is LDL located? Kind of in circulation, right? But it's very close to the epithelial wall, the capillary wall. What about HSL? I can't hear you. Somebody. Yes, adipose tissue, but in terms of inside or outside cells? Inside of cells, right? Okay, so HSL is localized inside of cells. And what about the function of these two enzymes? Are they same, similar? Yeah.
So LDL breaks them down to bring them into the cell, and then HSL breaks them down within the cell to export.
Release, yes, excellent. So they carry the same reaction. They hydrolyze triglyceride, right, and break down triglyceride to fatty acids, cholesterol, but the substrate is different. And the HSL basically break down the triglyceride inside of cells, particularly in adipose cells, right? And break down to the fatty acids, and this fatty acid being released and out of cell into the circulation, right, during the fasting condition. But LPL break down triglyceride inside of lipoproteins, right, chylomicron and the VLDL, and in the circulation. So it's different substrate, although they catalyze the same reactions, right? But other thing is the regulatory mechanism is also different. So imagine the LPL in the adipose tissue is activated, right, during the fat condition, stimulated by insulin, right? But HSL, what you expect HSL activity would be high or activated? Fasting, I saw you say fasting, thank you. Fasting condition, right? So insulin basically suppresses HSL So the difference, right? Yes.
Did you say that LDL is active though during some inflammation from the muscle?
Yeah, we're right. So the muscle is another issue, but here we talk about adipose tissue, right? And we also make a comparison, right? The LPL between adipose tissue and muscle, right? So don't be confused, right? There's two enzyme, HSL, LPL in the adipose tissue. Make comparison, and they have a different location and catalyze the different substrates, right? Substrate in different location, and the purpose is also different, right? One the pickup, for pickup of fatty acids, and the other one's the release of fatty acids, right? Okay. All right, so let's move on to lipid metabolism 7. Regulation of lipid metabolism, and I'm also going to talk about some clinical relevance, right? And just go over again the 5 different lipoproteins, like the composition of the lipids, and quite different. And chylomicron, VLDL are the triglyceride rich, right? And so you can see the percentage or proportion of triglyceride is huge, right? Particularly chylomicron is compared to other lipoproteins. And HDL, LDL are the cholesterol rich, right? So you have lots of cholesterol esters in the LDL particularly, and also HDL. Okay, so here I just give sort of summary of the lipoprotein metabolism. I put all the lipoprotein together and in one slide, and hopefully it's not too complicated. But at this slide, that basically will show something happening in the fat condition, right? In the fat condition. So let's start with the diet, right? When we eat and we absorb lots of lipids, including triglyceride, cholesterol, and we also get glucose, right, from the diet. So chylomicron is synthesized, we all know, in the small intestine, right, and pack the triglyceride and cholesterol. So chylomicron, the purpose, the function of chylomicron is to transport lipids from small intestine to the other tissues, right? The primary tissue that the chylomicron is transport lipid to is adipose tissue and liver, right? So chylomicron, first of all, synthesized is nascent, right, from small intestine, and receive the additional apoproteins. We didn't present here, but you guys know, right? And then become mature chylomicron. So chylomicron, once it reach the adipose tissue, and lipoprotein lipase, LPL, is released from adipose tissue, adipose side, can hydrolyze the triglyceride within the chylomicron, right? Release fatty acids, and these fatty acids picked up by adipose tissue and re-acidified back to triglyceride and stored in the adipose tissue, right? See? And the remnant, so once they finish this process, pick up by adipose tissue, and they still have some triglyceride left in the chylomicron, and you can see the significant decrease the triglyceride content if you come in the chylomicron remnant compared to mature chylomicron, right? But this chylomicron remnant eventually being picked up by the liver, and the apoprotein on the chylomicron is important for this pickup, right? Can you tell me which one? ApoE, yeah, I think everybody knows, right? ApoE is important, right, for this pickup of chylomicron remnant. Okay, so this is the chylomicron. And then in the meantime, the VLDL is also involved to transport the triglyceride to the adipose tissue in the fat condition, right? So you can see here, and the triglyceride is also synthesized, right, from two different sources. One is, from chymicron remnant. Chymicron remnant, still liver pick up triglyceride, which has not been completely picked up by the adipose tissue, right, and then go to the liver. So liver picks the remaining triglyceride from chymicron remnant, and also liver uptakes glucose, right, and glucose can be converted to fatty acids through lipogenesis, de novo lipogenesis pathway, right, and fatty acids, being acidified to triglyceride. So this triglyceride, the fatty acid in this triglyceride basically from diet, from chymicron remnant, and from the de novo lipogenesis from the glucose, right? And this triglyceride being packed to the VLDL, right? So VLDL and is transported to adipose tissue. And again, the LPL, hydrolyzed triglyceride, release fatty acids, is also pick up, right? So VLDL in this case serves sort of a backup mechanism, to make sure that all triglyceride being transported to adipose tissue for storage in the fat condition, right? And here I actually did not put the other pathways during the fasting. VLDL also can be synthesized in the liver, right? Yeah, question.
What do the different colors mean?
Sorry, colors?
Yeah.
Which?
Like the blue and then the red and black arrows, what do they indicate?
Oh, this. I just try to. You guys recognize the blue means the transporter. The pathway of VLDL, IDL, LDL, right? And black, represent the chymicron pathway. I don't think it's really mean anything, but just try to, you guys, being able to recognize, right? Too many arrows here, right? So, okay. Yeah, maybe a little bit, maybe this like HDL, right? And let's see, VLDL, IDL. You can maybe just follow the color, right? Okay. I got lost. Oh, here. So, the other pathway in the fasting condition, right? And I did not put this pathway. The VLDL, the fatty acids mostly picked up from the circulation, right? Fasting condition that the lipolysis is being stimulated and a lot of fatty acids being released from lipolysis of adipose tissue and go to the liver and synthesize triglyceride, right, and pack to the VLDL. And this VLDL in the fasting condition is not actually much transported to adipose tissue, but to the skeletal muscle, right? And skeletal muscle release LPL. Remember, fasting condition, LPL in skeletal muscle is increased, right? And hydrolyze triglyceride, release fatty acids. And again, the fate of fatty acids released from VLDL is mostly being utilized for oxidation in the skeletal muscle for energy during the fasting, right? Okay, now let's look at the VLDL, right? So ideal VLDL basically is related to the VLDL, right? Because it's sort of a remnant of VLDL, IDL. So once the VLDL triglyceride being picked up by adipose tissue, then VLDL become the remnant, which is IDL, right? So you see, and you have a less triglyceride because a lot of triglyceride being picked up by the adipose tissue. So IDL then become the LDL. So IDL, most of IDL actually is picked up by the liver, right? So, and then you lose a lot of triglyceride in IDL and become the LDL. And LDL, if you can see that the cholesterol ester content is higher compared to IDL. So where this cholesterol esters come from in the LDL? Yeah, from HDL, right? So there is called a lipid exchange process happening in the circulation. So LDL and receives lots of cholesterol esters from HDL and gives the some triglyceride back to the HDL, right? So LDL then become cholesterol ester rich lipoprotein, and again most of the LDL being picked up by the liver, and the reason is because liver has the higher content of LDL receptors, right? So IDL and LDL, they are all picked up by the liver through the LDL receptor mediated mechanism. So LDL receptor are essential for the pickup of IDL and LDL, right? And some of the LDLs also, go to pick up by the peripheral tissues, including macrophages in artery wall, right? And lastly, the HDL. We know we just talked about it, right? HDL beta HDL initially synthesized from the liver and the lipid pool, HDL, and they go to the peripheral tissue, pick up free cholesterol and phospholipids, and then become lipid-rich alpha HDL, right? And also several two steps that occurs in the plasma. Do you guys still remember? Acidification, right? So the free cholesterol has to be acidified to cholesterol esters that occur in the plasma in the circulation, and then eventually and also exchange the lipids between these three lipoproteins, right? So alpha HDL eventually become cholesterol rich and also get triglyceride from VLDL, IDL, LDL, right? And alpha HDL eventually goes to the liver, picked up by the liver, and through the receptor. Remember this receptor that pick up alpha HDL is a scavenging receptor, right? HRB1 receptors in the liver, right? Any questions about this whole process? The all different 4 different type of lipoprotein that involve the transport of triglyceride and cholesterol, right? Yes. You mean LDL? Yeah, LDL has the B100. So both ApoE and ApoB 100 can be recognized by LDL receptors, right? Other questions? Okay. All right, so regulation of the lipoproteins, and we already mentioned, right, many times that the chylomicron and VLDL are cholesterol, triglyceride-rich lipoproteins, and primarily regulates the triglyceride transport and metabolism, right? So these two lipoproteins are very important in the regulation of cholesterol levels in the circulation. And LDL, HDL, basically the cholesterol rich, right, lipoproteins, and plays most important in the regulation cholesterol homeostasis levels. And it also, these three lipoproteins also involve so-called triglyceride cholesterol ester exchange, right, in the circulation. And all these four different type of lipoproteins that involved in the regulation cholesterol homeostasis, although the LDL and HDL play the most important role, right? So the dyslipidemia, you guys know, right? So lipoprotein metabolism is very important. If any process is wrong, then will affect the lipids homeostasis, including so-called dyslipidemia, including increased triglyceride level or cholesterol level, in the circulation, and also increase the cholesterol level or deposition in the tissues, and causing a lot of metabolic problems, including obesity and cardiovascular disease, right? And in terms of regulation, the regulated factors can be genetic or dietary or environmental, right? So I think here we're going to talk about both genetic and dietary factors, and how they control or regulates the lipid metabolism, right? So in terms of a dietary fat, particularly composition of fatty acids, whether it's saturated fatty acids or unsaturated fatty acids, these are very important, and they usually influences the levels of cholesterol and the triglyceride in the circulation, right? And also dietary carbohydrate. So dietary carbohydrate, if we have a high carbohydrate diet, and usually also influence the triglyceride levels, and not much to do with the cholesterol levels, right? So in other words, the cholesterol and the triglyceride levels can be affected by dietary fat, and dietary carbohydrate seems to only affect the triglyceride levels in the circulation, right? They're just in general, right? In general speaking. And specifically, the factors that affect the plasma lipids, first of all, is the percentage of total calories consumed as a fat. So, we have a different composition, right? In the diet, we have, we need the protein, fat, and carbohydrate, but in the proportion, right? In right proportion. But if we have too much fat in the diet, that will affect definitely the plasma lipids levels. And the second thing is that the proportion of saturated versus unsaturated fat. So saturated fat, we all know, is bad, right? The bad lipids. So if we have too much saturated fat in the diet, and it also affect the lipids level in the circulation. And last thing is amount of dietary cholesterol. That we know that 50% of our body cholesterol from diet, and another 50 from the synthesized, right, in the body. So dietary carbohydrate, the cholesterol definitely will affect the cholesterol levels in the body. And the major effect, and again, just usually affecting the circulating triglyceride and the LDL cholesterol levels, right? Now let's talk about the dietary sources of energy. And average versus desirable. The average means that in the American population, that the average, how much we take, consume the different nutrients, the percentage, right? So average, like we have about a 42% carbohydrate in our diet, and 40% of fat, and 15% protein, and 3% alcohol that we consume on average, right? And the desirable, so if you compare this, like desirable, we wanted to have a carbohydrate at 55%, and fat is 30%, and protein it's okay. I mean, we probably have maybe too much fat, right, in our diet, and also alcohol. We consume too much alcohol. And fat, in terms of the little bit more detailed, the fat and saturated. We have 17% of saturated fat in the diet, and 12 monounsaturated percent, and 6% polyunsaturated, and 2% trans fat. And let's see the desirable composition of fat in the diet. We wish we will have less, like 10% of saturated, and we're okay with the monounsaturated and polyunsaturated fatty acid, but we have a little bit too much trans fat, right, in the diet. So, and a carbohydrate, have starch 23, and sucrose 14, and lactose 3, and glucose 2%. And let's see, desirable, we wish we will have more carbohydrate from starch, and less from sucrose, right? And glucose, lactose, it's fine. So now let's go back to the VLDL regulation of synthesis and secretion VLDL. And we talk about this, the sources of fatty acids used for synthesis of triglyceride in VLDL in liver. We have talked about a lot, right? So you guys all know, and one is from de novo, and lipogenesis from carbohydrate. If particularly when we eat a high carbohydrate diet, and uptake of free fatty acids from the circulation, right? One can from the diet, right, and the other, during the starvation from triglyceride storage breakdown, and the diabetic condition, right, you also have a lot of fatty acids. And enhance of the synthesis of secretion, and in which kind of condition that VLDL synthesizes increase. So one is if we consume the high carbohydrate diet, right? So we have lots of de novo lipogenesis happening, and then we synthesize a lot of fatty acids in the liver. So this fatty acid need to be transported to the adipose tissue. So that makes the liver to synthesize more VLDL to pack the for transporting fatty acids, which synthesize from the carbohydrate to the adipose tissue. So this is one condition that VLDL synthesis and secretion can be stimulated and increased. And a second condition, like high levels of circulating fatty acids. One is we know that under fasting condition, right? So your liver picks up a lot of fatty acids from circulation. And other condition is the diabetic condition, right? So you have a lot of fatty acids in the circulation. So this will also need the VLDL to transport them to the site that utilize fatty acid, for example, in the muscle in the fasting conditions. And the diabetic condition is kind of pretty different from the normal physiological fasting conditions. And the diabetic condition, you have a lot of fatty acids being picked up by liver and synthesized triglyceride. And usually you also have some problem with the VLDL secretion, right? So that means this triglyceride, the fatty acid pickup from the circulation mostly deposit in the liver, causing fatty liver disease under the diabetic condition, right? And then the injection of the alcohol, in this condition you have a lot of fatty acids also synthesized in the liver, right? At high concentration of insulin and low concentration glucagon, which is under the actually fasting condition, right? So you can increase the fatty acid release. We already talked about it. So, any questions about this regulation of VLDL? Right. So I just wanted to summarize here, make it a little bit more clear. So if we ask you a question, in which condition that VLDL will be synthesized would increase. One is high carbohydrate diet, high fat diet, right? You increase the circulating fatty acid levels, and also during the fasting, right? These are the physiological conditions, right? Okay. Fatty liver disease. So fatty liver disease means that the triglyceride, a large number of triglyceride it's accumulated in the liver, right? So this can be caused by imbalance between triglyceride formation or synthesis and the triglyceride export, means by secretion through the VLDL, right? So if the balance, for example, either due to the synthesize too much triglyceride in the liver, or you have some problem to export the triglyceride out of the liver, this imbalance could cause the accumulation of triglyceride in the liver and eventually develop a fatty liver disease, right? So what are the possible defects that can cause fatty liver? We know the general causes, right? Is imbalance, synthesis too much or exported less. It's the main main reason. So what are the possible defects that can cause fatty liver disease? So fatty liver disease, here we actually two different types of fatty liver. One is called non-alcoholic, and the other is the alcoholic. So non-alcoholic means that it's caused by the reasons are not related to the alcoholism, right? So two types of non-alcoholic fatty liver. So first type of fatty liver can be caused by increased levels of plasma-free fatty acids. For example, diabetes, I mentioned before, or chronic intake of high-fat diet. If you always eat high-fat diet for many years, then you probably will develop fatty liver disease, right? So this is the first cause. And the second type of fatty liver disease, non-alcoholic fatty liver disease, is some metabolic blocks of the production lipoproteins, right? In other words, that lipoprotein cannot be made, right, in the liver, so cannot transport triglyceride out of the liver. So this cause usually related to the genetic, problems, right? You either missing some, apoproteins or some other the phospholipids. So then lipoprotein cannot be made, right? So for example, block in the apple protein synthesis, we know apoprotein is very important, right, to make a structure of a lipoprotein and to stabilize lipoprotein. So if some problem with apoprotein synthesis or lipoprotein lipids, for example, the phospholipids cannot be provided for some genetic reasons, or impaired the secretion of lipoprotein. So missing some part of the structure component of lipoprotein, so lipoprotein cannot be secreted. So all this reason more related to the genetic problems, and then can causing the problem with the formation of or synthesis of lipoproteins, right? Okay, so do we have informal writing today, right?